So let's now construct genetic map based on the 3-point test cross using a test cross with fruit flies. So let's imagine that we are the geneticists and we perform test cross with a fruit fly which is wild type for eye color, body color, and bristles. Of course, if you want to construct gene map, the fruit flies has to be heterozygous for all three genes. So in eye color, red eyes are dominant over scarlet. In body color, gray is dominant over ebony. And in bristles, normal are dominant over small bristles. So you perform crosses with wild type heterozygous fruit flies. Remember, test cross, the tester is always homozygous recessive. So afterwards, you obtain your progeny and you count 755 individuals and you get 8 different phenotypic categories. So based on what you know about these genes, you can assign for each phenotype the genotypes. They are outlined right here. Now, when you want to construct gene maps based on 3-point test cross, there are certain steps you have to follow. First step is to identify non-recombinant progeny. How do you identify them? There are two most numerous phenotypes. So which ones they are here? Wild type and all mutant. So you know that these two are non-recombinant because they are most numerous. In second step, you identify the double crossover progeny which is a result of double crossing over. And how do you define them? They are two least numerous phenotypes. So the least numerous phenotypes is this one, spineless and scarlet ebony. So these are the double crossing over. So these are double crossing over and these are non-recombinant. So once you identify these two categories, non-recombinant and recombinant, which underwent double crossing over, you can define the position of the three genes on the chromosome. So you can define which gene is in the middle, and we discussed it in previous recording. So which gene is in the middle? You remember, you compare genotype of double crossover to non-recombinant. So if you look in non-recombinant, of course, these recessive alleles were obtained or received from tester. So the non-recombinant position genotypes are ST wild type, E wild type, and SS. So all three wild type or all three mutated. So these are non-recombinant genotype which were in gamete obtained from wild type fruit fly. So now let's compare it to double recombinant. So in this double recombinant, you see you have wild type ST, wild type E, which resembles this non-recombinant, and what is different? It is SS, which is different here. Here you have mutated SS. Let's compare second double recombinant. So you have mutated ST, mutated E, so it resembles here. However, you have wild type SS. So again, SS is different. So what does it tell you? That SS is in the middle. So SS, which defines the bristles, is in the middle. So position of this gene is ST, SS, and E. Okay. Now, what you want to define here is which gene is in the middle, and we know that based on the comparison of non-recombinant with double crossed over individual progeny, you know that SS is in the middle. Which gene, whether is it ST, whether is it gene for eye color and gene for body color, which one of them is on 5 prime, you cannot define. It can be ST as SE or it can be E as ST. If you want to define proper orientation regarding to 5 prime end of the chromosome, you have to perform additional test cross and with additional genes in order to define proper orientation. Would you can define is gene which is in the middle, and you know that it's SS. Now, if you are not sure how to, if you forget how to define it by the simple way, we will discuss additional way how you can do it in the next slide. If you are not really sure which gene is in the middle, you can actually outline a position order of the gene that each of the 3 genes will be in the middle. So we outline 3 different scenarios. Under first scenario, you have gene for eye color in the middle. Under second scenario, you have gene for body color in the middle, and in third one, you have gene for bristles in the middle. Now, under each condition, you outline what would happen after double crossover right here, and you see afterwards you will have an individual which underwent double crossing over. So let's look at the first scenario. So the gamete of this individual will be this one and this one. This would be 2 gametes of under first scenario. So the double crossing over individuals, offsprings, meaning the least frequent offsprings, will have gray body, scarlet eyes, and normal bristle. And second least frequent category would be ebony body, red eyes, and small bristles. If you look now in the table 7.13, you will see that these offsprings are not least numerous. Actually, these are intermediate. This group of individual has intermediate number. It is 52 and 50. So this is not double crossing over. So this is not situation which happen. And you proceed this way with second one and third one, and you will find out that actually it is a third situation which actually correspond phenotypically to least numbered groups of offsprings, meaning that this is the position of genes on the chromosome. So once you determine position, meaning which gene is in the middle, you can calculate recombination frequencies and you can construct genetic map. So what you have to do first, you have to calculate recombination frequency between genes STSS, and afterwards you calculate recombination frequency between genes SS and E. And how do you do calculation? You remember recombination frequencies we calculate as the ratio of recombinant divided by total number and multiplied by 100%. So for STSS recombinant, you go again to table 7.13 and look for recombinant, single recombinant of between gene STSS, which you will find is 50 plus 52, and you add also, because double recombinant, be careful, double recombinant also comprise single recombinant between these 2 genes, so you have to add also double recombinant. You divide by total number and you see that the percentage of recombination is 14.6%, so distance between ST and SS is 14.6 map units. You do the same for second recombinants between SS and E. So here you have recombination frequencies between SS and E is 43 and 41, and again you have to include double recombinant, and you proceed the same way, divided by total number, and you find the recombination frequency is 12.2. So distance between SS and E is 12.2 map units. So if you add it together, so distance between ST and E will be 26.8 map units. So if we summarize, if you do 3-point test cross, what do you have to do? Firstly, you have to perform experiment and count the offspring. Count every single phenotype category, number assigned, number of individual to each phenotypic category. Afterwards, you have to identify the non-recombinant progeny. Secondly, identify the double crossover progeny, and by comparing phenotype of double crossover progeny with phenotype of non-recombinant, basically they should be alike except one characteristic. They should be alike in two characteristics and differ in one. So if you look in genotypes, you will find which gene is different, and you will find out that the gene which is different is in the middle. Once you find out the order, you find out which gene is in the middle, you calculate recombination frequencies for each two genes, you first two genes, second two genes, and since double crossover also includes single crossover, you have to include those in the calculation. Next thing I would like to point your attention at is interference and coefficient of coincidence. Why is that? Let's go back to our example. In theory, double crossing over gametes with double crossing over should be equal to the single crossover multiplication of single crossing over. So basically, if you want to calculate probability of double crossing over, in theory, you should use multiplication rule of probability and multiply probability of have single recombinant between ST and SS, which would be 0.146, by probability of another single recombinant, which would be between SS and E, which would be 0.122, and multiplied by total number of progeny, which was, as you recall, 755. So this multiplication will yield the number 13.4. So this is what you would expect if two crossing overs between gene STSS and gene SS and E were independent of each other. However, as you recall, we only observed 8 double crossover, 5 plus 3. So why is it? Basically, when you have 3 genes and crossing over occurs between first two, it will interfere with second crossing over between second and third gene, and the degree to which this one first crossing over or crossover interfere with additional one we call interference. To calculate interference, basically first you have to calculate coefficient of coincidence right here. Coefficient of coincidence actually is division. You divide number of observed double crossing overs, which was 8, by number of expected, so we expected 13.4. So coefficient of coincidence in this case will be 0.6. So what does it mean? That actually we observed only 60% of expected double crossovers. Afterwards, once you have coefficient of coincidence, you can calculate interference as 1 n 1 minus coefficient of coincidence. So interference will be 1 minus coefficient of coincidence is 1 minus 0.6, mean is 0.4, which indicates that 40%, from here it's 40% of double crossover progeny expected. So 40%, which we expect, 40% out of expected double crossover will not be observed because of interference. Finally, I would like you to realize that up to now we only talk about crossing over between two non-sister chromatids. However, during one meiosis there might be multiple crossover, and many of them can go undetected, and therefore the progeny of this crossover based on distance, based on this undetected crossing over, the distance we determine by the genetic map based on the recombination, underestimate real true physical distance between genes. So if you really want to know true physical distance, you have to construct physical maps.